Hello, my name is Nobu Acevedo, and for my Chem 394 final project, I am proposing steps for investigation of BmK AGAP analgesic properties through molecular dynamics simulations. This is Buthus martensi karsh, commonly known as the Chinese scorpion. It's a species of scorpion found in China, Mongolia, and Japan that prefers dry areas and grows up to 2.4 inches long. I'll be addressing it as BmK for the rest of the presentation. BmK has been used in traditional Chinese medicine for thousands of years to treat various diseases like epilepsy, facial paralysis, and apoplexy, and has been used as an analgesic with relative success. The claims can be exaggerated, but recent characterization of the toxin and peptide structure has paved the way for continual research and possible use as a treatment mechanism against cancer cells. Scorpion alpha toxins are a single-chain polypeptide consisting of 60 to 72 amino acids and can be divided into 3 subfamilies according to their activities against mammals and insects. There's the classical alpha toxin, which is an anti-mammalian toxin, highly toxic to mammals and weakly active towards insects. There's the insect alpha toxin, as the name states, is a toxin that is extremely active towards insects. And then we have alpha-like toxins, such as toxins from BmK, that can act on both insects and mammals. Most scorpion alpha toxins have a common framework consisting of 3 double-stranded anti-parallel beta sheets, shown in yellow, as beta strand 1, 2, and 3 are linked to a short alpha helix, as you can see in red in the figure. The loops are listed in green. The functional surface of these scorpion toxins is separated into 2 domains: the non-core domain, consisting of the 5 residue turn of residues 8 through 12, and the C-terminal segment, residues 56 to 64, depending on the toxin, and the core domain of the residues in the loops preceding the alpha helix, between the beta-2 and the beta-3 sheets. Of particular interest is the non-core domain, which is considered to determine the toxin specificity due to its unique tertiary arrangement. In one study, protein BmK AGAP SYPU2 was isolated and purified from BmK venom and was shown to have analgesic activity at the animal level. Researchers from Shenyang Pharmaceutical University and Jilin University were able to determine the C-terminal segment of the non-core domain help maintain its protruding topology, which is associated with analgesic activity. By deleting the glycine 65 and glycine 66 in the C-terminal segment, the molecular dynamics simulation showed that the protruding topology is significantly disrupted and made flat. And the figures here show that BmK and its wild type, and then the mutated proteins where glycine 66 was deleted in the center, and glycine 65 and 66 were deleted. And you can see that the protruding topology gets flatter and flatter with the reduction of the glycine residues. In January 2019, researchers published on the effectiveness of BmK venom's anti-tumor analgesic peptide, also known as AGAP, against breast cancer cells. The human breast cells were gathered from 36 females diagnosed with varying grades of breast cancer, from grade 1 to grade 3, confirmed through histopathology analysis, and the 42 females from a control group with no history of breast cancer. The breast cells were first examined for pentraxin-3, or PTX3 expression. This is a modulator of the inflammation process and plays a role in the activation of complements and the regulation of inflammation. Pentraxin-3 is regulated by nuclear factor kappa B and tumor necrosis factor alpha. Activation of kappa B signaling promotes cancer cell growth and chemoresistance, while tumor necrosis factor alpha is a protein that promotes pentraxin 3. Breast cancer cells have been found to have an overexpression of pentraxin 3, and although the exact role of pentraxin 3 in breast cancer remains unclear, the data shows that pentraxin 3 is associated with stemness, higher metastatic potential, and epithelial mesenchymal transition. Epithelial mesenchymal transition is a process that allows epithelial cells to undergo changes that make it assume a migratory status, and it has elevated resistance to apoptosis as well. So after treating the in vitro breast cells with AGAP, it was discovered that BmK AGAP inhibited the breast cancer cell stemness, EMT, migration, and invasion by downregulating pentraxin 3. The same result was noted with in vivo testing of mice with breast cell xenograft tumors. But how does BmK do this? BmK AGAP has been reported to block neuron transmission by binding to voltage-gated sodium channels and inactivating them. These sodium channels are a crucial mechanism for pentraxin 3 because tumor necrosis factor alpha uses the channels by increasing the voltage-gated sodium current and increases the expression of the channels through nuclear factor kappa B pathways. The results showed that BmK AGAP decreases NaV1.5 expression, which is a transmembrane protein associated with the regulation of nuclear factor kappa B by inhibiting voltage gate sodium channels, thus decreasing overall PTX3 expression and inhibiting cancer cell growth and epithelial mesenchymal transition. So why use BmK peptides instead of current opioid analgesics? Are there any benefits? Why do it in the first place or spend money? Well, there are currently some reports indicating that morphine and fentanyl, which are common opioids used to treat cancer patients, may promote cancer cell stemness, epithelial mesenchymal transition, and chemoresistance. One study by University of North Carolina, looked at the survival rates of an earlier study of 2,000 breast cancer patients, found that women being treated for invasive breast cancer who had genetic mutations that made them less sensitive to opioids were much more likely to be alive 10 years after cancer treatment. Women with one copy of the protective mutation were nearly twice as likely to have survived. Those with two copies were 4 times as likely. Another study by the University of Medicine Chicago found that various types of human lung cancers have 5 to 10 times as many opioid receptors as non-cancerous lung cells. These cancer cells that had additional copies grew more than twice as fast as tumor cells that lacked extra receptors when transplanted into mice and were 20 times more likely to spread to distant sites. Medication blocking the opioid receptors reduced the tumor growth and spread. Albeit a extremely low percentage of average patients, there is also the possibility of substance abuse or addiction in patients. One study recorded 4 cases of addiction out of approximately 12,000 inpatients prescribed opioids. This is obviously an extremely low percentage. However, no data has been released as of yet of opioid addiction rates of patients who do have personal or family history of substance abuse or addiction. This is likely to be a much higher number. So the presence of BmK analgesics or anti-tumor related drugs or treatments would enable patients who may have complications with opioids or history with substance abuse another option. So who's currently working on it? The current researchers are primarily from these 4 institutions. There are others. However, the mainstay of the research has been provided by Shenyang Pharmaceutical University in Shenyang, China, Dalian Medical University in Dalian, China, University for Development Studies in Tamale, Ghana, and then Jilin University in Chengchun, China. And to note, there's been no Nobel Prize awarded in the research area as of yet. So here are my proposed steps for further investigation of the correlation between the analgesic properties identified in the non-core domain of BmK AGAP. And my step proposals for the 5 residue turn, and see what effects may happen from either disrupting or changing or mutating the 5 residue turn, similar to what was done with the C-terminal end. So using simulated mutations of the residues and then conducting molecular dynamic simulations of said mutations and the wild type through NAMD, and then comparing and contrasting these the RMSD plots and the RMSF plots between the mutations and the wild type. So the first thing that needs to be done is to download the PDB file from the RCSB website. And I have done this. The wild type that I downloaded was called 2KBH. But before I can do any dynamic simulations, first I have to build a protein structure file. So this can be done by going into extensions in VMD, modeling, and then the automatic PSF builder. Once you're there, you will need to download the topology files from, there are a number of websites that they can be downloaded from, but most of them are listed in the CHM wiki websites. But once that's done, I had no other issues, and I was able to generate an automated PSF file. Okay, so once you have the PSF file and PDB, you can use this to mutate a residue of choice. And I did this through VMD. There's a residue mutator, as long as you have the PSF as shown in the slide, and the PDB, you can choose a residue, whichever one you want to mutate. And as long as you list the number here and the ID of the target residue, and you mutate it to whichever 3-letter mutation you'd like, and you can run the mutator, and it will generate a new PDB. So the mutation that I chose was aspartic acid. I mutated this into alanine to observe the effects of hydrogen bond disruption. So as you can see here in the wild type listed from the RCSB website, there is the presence of a charge-charge interaction between aspartic acid and cysteine, which is about 2.92 amperes, and then hydrogen bonds between cysteine and aspartic acid, and then asparagine and aspartic acid, and these are listed as to 3.23 amperes and 3.50 amperes, respectively. So once this was done, I had the new PDB file with the mutated protein, and I created a new PSF file. With both files ready, I ran the measure min-max command on both proteins to calculate their X, Y, Z center points and then their sizes as well. So once that is done, I can have all that information prepared and they're ready for their molecular dynamics simulations. I also use Avogadro to prepare the molecules for Gaussian analysis. After loading the wild type and the mutated protein in Avogadro, I optimized the geometry of these molecules, which can be done just by going to extensions and geometry optimization. And then after this was completed, I prepared the .com file for energy analysis in the Gaussian view. And with that, I was ready to analyze all the files. For issues and troubleshooting, I may have been a little too hopeful about the configuration file and the .com files that I created. The first issue I ran into was when I created the configuration file, I was given a fatal error, and this was the listed fatal error for it. And that was resolved just by recreating the configuration file. And then after I tried to run it again, there was another fatal error in that the NAMD was unable to find the van der Waals parameter for atom type HB1. So this is likely to be a topology file recognition issue. I'm not sure which topology file wasn't recognizing and why it wasn't recognizing it. I did download all of them, and I did test some of them beforehand, but it remains unresolved. So I was not able to conduct the molecular dynamics simulations. And then the .com files that I attempted to run on Gaussian ran into a memory issue. The required amount of memory as given was 120, and I attempted to go into link 0 and run this command, but I was unsuccessful. So that remains unresolved as well. So proposed next steps from the configuration file and the .com file that has been created. As long as all issues are resolved, you could just go forth and do the simulated mutations, simulated molecular dynamics of the mutated residue and the wild type in the 5 residue turn, and how that affects the possible conformation and any other bonding interactions. You can also measure the bond length, angle, the torsion properties, and then the energy values of the mutated residue and the wild type in the .com file. You can actually just measure the bond length and angle and torsion properties without having to go into the .com file. However, the specific residues aren't identified, so you would have to kind of correlate those two with using VMD and Avogadro. And then once all that is done, you can compare and contrast the RMSD and RMSF plots between the mutations and the wild type for effects on topology, similar to what was done with the C-terminal segment. And these are all my references. And that concludes my presentation. Thank you for watching.